about 3 years now, and it seems like there is no need to give overview of CHAMP disease. I go straight to the point. We're working both in vivo, which is mouse model, and in vitro, which are cell lines, and I will give a little bit more information about it in next slides. So major focus at this moment is mouse study. We bought mouse model from University of California when it was generated, and it's having specific human mutation of 35 base pair. So we able easily, So we were able to easily genotype mouse model. We have another presentation following after mine when we have like all details of those phenotyping and explaining mouse model. We're also working, our hypothesis is the gene therapy using adeno-associated virus would be effective therapy for CHAMP1 disorders. Because gene, it's monogenic disease, and gene is just like sufficiently large, so it could be packaged into AAV. AAV have been around and showed efficient for multiple other diseases when they were used. And we are going to evaluate therapeutic effects of AAV using our mouse model. Also, in the same model, we will estimate therapeutic toxicity and the toxicity index by titrating doses of AAV vectors. We have sufficient amount of those AAV on hands and just perform first treatment. And in about 8 weeks, we're going to be able to start testing the mouse model. As to cell culture studies, first, we were using this SH-SY5Y cells, which are neuroblastoma cell lines. They've been around for over 50 years and commonly used as a model. What we've done, we used the CRISP-Cas9 system to take pool of cells and generate mutation. This technology doesn't have we don't generate specific mutation, but it's like pool of cells with multiple different mutation. Some cells don't have any mutations. So we decided to perform what calls limiting dilutions. So we titrating cells, diluting them, and trying to put just one cell into 96 well plate. This grows for several weeks. Not all clones are viable, but the one we obtain, we sequence and determine if they have mutation or if it's cells like wild type. After that, we were able to find one clone with full absence of CHAMP1 expression. So it's a good model to work and provide, we were able to confirm it both on RNA and protein level. And next, we treated that cell line with AAV, CHAMP1, AAV type 9, exactly the same AAV we will use in mouse model. And we were successful in terms that mRNA expression is increased and stayed upregulated. Proteins, you can see first one is wild type, then clone with no CHAMP1 expression, and then expression comes back after treatment and stays detectable for whole experiment duration. So we consider it like good proof of principle. Next, recently we got RAN cells. It's human neuronal progenitor cell line. We perform the same procedure using CRISPR-Cas9 to generate mutation. On first blot, you can see that this is bulk experiment, all cells were obtained after CRISPR. There is a significant reduction in CHAMP1 expression, about 70%, but we went ahead and did those specific limiting dilution to generate several clones. We were able to get like about 30 clones. You can see on other example, it's just like some clones last as control, others have multiple bands, which just tells us that they have different mutations. Probably some in frame, some benign. We are sequencing them right now, and after we establish mutation, we will perform cell culture studies for clones which have very low or no expression of CHAMP. Advantage of these cell lines, it's like relatively new. It was generated just in early 2000s, and these cells have ability to differentiate into neurons and glia. So we will be able to do like more functional studies and probably some microscopy study to see differences between wild type and different mutation. So in conclusion, I just want to say, spoiler for next presentation, our mice model has phenotype, which in this area of research, it's already a good sign. So it's suitable for gene therapy studies. We also will continue work in cell lines because they are a good model to study mechanism and toxicity, drug testing. We are planning to work with iPS cell lines, which we haven't done yet, but iPS is very valuable when you develop in patient-specific models. My next slide was thank you slides. Thank you so much for your attention, and yeah, we're gonna keep working, so thank you.
Thank you so much. Any question?
Sorry if I missed it, but when you said that the mice have the phenotype, do you mind describing that really fast?
So next presentation was like 30 or so minutes about just mice.
Okay, great, thank you.
Hi. I don't know if you or anyone else in the room would know this, but the work you're doing with AAV9 on CHAMP1, would that not translate to POC-Z because it's a bigger gene?
Yeah, like unfortunately in AAV you can only load like relatively smaller genes up to 5 kilobyte. If gene is bigger, it needs to be truncated, but then it could lose functionality.
Do you know if there's like similar technologies on the horizon that could do?
I know that there is other viruses like lentivirus, adenovirus, which allow to package bigger genes.
Thank you.
I just want to ask a question about the virus expression. We're trying to do something similar for a different gene. One of the things that we're always thinking about is trying not to overexpress it, because sometimes too much is as much a problem as too little. Do you have an idea yet whether the AAV9 is expressing similar to the wild type levels?
We don't have data on animal models yet. As I said, we injected mice pups when they were like 3 days old, but to start functional studies, we are waiting till they 8 weeks, so it's kind of in a process. It will be done like this year, but no result yet.
But in the cell lines, is it?
So like,
how does it compare?
And actually in cell lines, we went as long as 72 hours, but I know from literature and from working on other diseases, I was working on Duchenne like long time ago. So in vivo models, like using an animal, AAV always works better and it stays on longer, almost like for mice, it's like for duration of their life. In cell lines, AAV doesn't have like longevity, like for long plastic cell lines. So when we done in neuroblastoma, they don't live that long on plastic. So we don't know, like 72 hours, kind of as long as you go with neuroblastoma cell line. But again, AAV should be better in vivo than in vitro. So if it works in vitro, we have high hopes, but we don't have like data about dosage.
Are you using a pan promoter? Are you trying to express it in all cell types? Or are you
Correct.
using a
Yeah,
neuronal
we're
promoter?
using, yeah.
Okay. The other just quick suggestion and to think about is that AV9 has a little more trouble passing blood-brain barrier in humans and primates compared to mice. You might want to think about serotyping it as a PHP-EB instead, which has looks like it has better transmission and higher vertebrates. And in fact, I think there's even a newer generation that hasn't quite hit the market yet that'll be probably game-changing for human.
Okay, thank you so much.
I might have a two-part question. For the folks that are non-medical, non-research in the room, can you please further explain what you mean by no CHAMP1 expression after using what you're using?
So we were using that CRISPR technology to knock out protein expression. On purpose, we generate mutation to use it as a model. And in some mutation, like it could be reduced expression, or it could be in frame deletion, so it's like keeps the same mutation. You can have deletion like 30 amino acid is still expression. So this mutation was that mutation completely shut down production of the protein. So like 100% absence.
So maybe it's a three-part question then. Can you also explain to us, it's my understanding as being a grandma of a CHAMP1 that the gene mutation in embryo is actually permanent. So how does after using what you're using change after birth? What we try, like we're not trying to, so what we're doing with this gene therapy project, we understand that like in human, it's already like one mutation and it's permanent. So we're using our mice, which is as a model. So it has one mutation, it's heterozygote. What we're going to do with AAV, we're bringing CHAMP1 protein in those AAV particles. And we're using cell culture, it's kind of like separate model. So with cell culture, we completely shut down protein, and we want to understand what's gonna happen in terms of like cellular processes. So maybe it could suggest some drugs. So it's like two separate kind of part of project.
Thank you very much.